So Opthea Limited is a public ASX-listed company. We're located in Melbourne, Australia, and our lead compound, OPT302, is a soluble form of VEGF receptor 3. It's essentially a trap molecule that blocks two alternative members of the VEGF family, VEGF-C and VEGF-D, and in doing so, it mediates neovascular effects, including the inhibition of angiogenesis and vascular permeability, as well as non-neovascular effects by affecting inflammation as well. Our lead program is in wet AMD, although this is a molecule that does have broad development potential across other eye diseases as well, including diabetic macular edema, and we're developing it as a combination therapy to be used with the existing VEGF-A inhibitors. In doing so, we are looking to achieve a much more complete targeting of the VEGF receptor pathway, and therefore to improve clinical outcomes for patients. Whilst we are complementary to VEGF-A inhibition, we are also potentially complementary to other products that are in development for wet AMD as well. And we have a phase 1/2A clinical trial that's ongoing at 14 clinical sites within the United States. So our long-term objective with this molecule is to address the incomplete responsiveness to VEGF-A inhibition. And this indeed may be due to the expression of other members of the VEGF family, notably VEGF-C and D, which signal through overlapping pathways with VEGF-A, but also through independent pathways to VEGF-A as well. Used in combination with a VEGF-A inhibitor, we can therefore achieve a much more complete blockade of this very important pathway that we know is involved in disease progression. In addition, we've got a phase 1/2A clinical trial that's ongoing. This is the design of that study. It is comprised of two parts. The first is a phase 1 dose escalation study consisting of 3 dose escalating cohorts of OPT302 dosed in combination with Lucentis, as well as a 5-patient monotherapy dose cohort of OPT302 dosed on its own. This phase 1 study has recruited 20 patients, and I'll go through that data in a moment. In addition, the phase 2A dose expansion trial is currently enrolling. We expect to enrol 30 patients into that phase 2A dose expansion trial. And each patient, the 50 patients in the phase 1/2A clinical trial, receives 3 monthly injections of our drug in combination with Lucentis or on its own over a 3-month period, with the primary data readout at week 12. We recruited 20 patients into the phase 1 study. Importantly, 17 out of 20 of those patients had occult lesions, with only 3 of the patients having lesions with a classic component. And importantly, 70% of the patients that we recruited were considered to be difficult to treat patients. These are patients that had previously received therapy with a VEGF-A inhibitor, but had shown a sub-responsiveness to VEGF-A inhibition. And 30% of the patients that we enrolled were treatment naive. OPT302 successfully met the primary safety objective in the phase 1 dose escalation study. There were no dose-limiting toxicities. We did not reach an MTD, and there was no evidence of drug-related immunogenicity. Overall, 16 out of 19 evaluable patients at week 12 maintained or gained vision from baseline, and no patient lost more than 3 letters. In terms of the combination patients, the patients that received combination OPT302 plus Lucentis, across all of the patients, both naive and prior treated, we recruited 14 such patients. There was a mean visual acuity gain from baseline at week 12 of 8 letters, and a mean decrease in central subfield thickness from baseline at week 12 of 91 microns. Looking only at the treatment-naive patients, of which there were 4 out of the 20 patients in the phase 1, the mean gain in visual acuity from baseline at week 12 was 16.5 letters, with a reduction in central subfield thickness of 214 microns. Looking only at the prior treated patients, of which there were 10 prior treated patients treated with the combination OPT302 plus Lucentis, the mean change in visual acuity from baseline in those patients was 4 letters. And in these patients, which had otherwise hit a ceiling effect of VEGF-A response, we showed a further reduction in central subfield thickness of a further 11%, or 42 microns, in this difficult-to-treat patient population. Taking a closer look at one particular prior treated patient, this is a patient that was treated with OPT302 plus Lucentis. This patient had received 6 prior doses of EYLEA combination therapy, and at baseline in our study presented with persistent retinal fluid, but after 3 months of therapy with the combination OPT302 with Lucentis, achieved almost complete resolution of that retinal fluid. In another patient, this is one treated with the combination therapy, again a prior treated patient that had received 10 prior administrations of a VEGF-A inhibitor. This patient experienced a very significant and a very encouraging improvement in visual acuity after 3 months of dosing on our combination therapy with OPT-302. In terms of the monotherapy arm, in terms of the phase 1 clinical part of this study, 3 out of the 5 patients received did not require rescue therapy, and all of those 3 patients that received the monotherapy actually gained visual acuity between 2 to 6 letters, which was quite important for us to identify a clinical signal with our molecule as a monotherapy with this drug. So just to summarise, in terms of our OPT-302 program, this is a program that addresses a large unmet medical need for wet AMD. The current treatments only target VEGF-A. We have a molecule, a trap molecule, that blocks 2 alternative members of the VEGF pathway, and these are members that are associated with incomplete responsiveness to VEGF-A inhibition. We've met the primary safety objective of our phase 1 clinical study, showing that OPT-302 is safe and very well tolerated, and we've shown early evidence of clinical activity. Patients that were treated with the combination of OPT-302 with Lucentis showed improvement in visual acuity, reductions in retinal thickness that were equal to or superior to week 12 data with Lucentis alone, despite us having recruited difficult-to-treat sub-responsive patient populations with a very high proportion of occult lesions. In the naive patients, our data suggests meaningful additive visual acuity gains, reductions in retinal thickness that were not expected with Lucentis alone, and in the prior treated patient population, with those patients that had otherwise hit a ceiling effect with VEGF-A inhibition, we showed evidence of a meaningful additional visual acuity gain and reductions in retinal thickness, suggestive that more complete blockade of the VEGF pathway can achieve clinical benefit in patients. The totality of our data is very encouraging. It does warrant us moving forward with a larger phase 2B randomised controlled clinical trial, and we're actively planning for that study in 2017 and accruing into our phase 2A clinical study, which we expect to report out in the first quarter of 2017. Thank you very much.